about my primary area of interest. So biohacking is a very broad thing, and some people want to optimize their health, their cognitive abilities, but many of biohackers, are aiming towards an anti-aging. We don't want to age, and I would like to start with the definition of the word aging. What does it mean? What is aging actually? And the most suitable definition, in my opinion, is the following thing. Aging is actually an increase of probability of death from any cause with advancing age. So you can see here on the graph, it's actually a logarithmic scale that with advancing age, our chance of death is increasing, and it is increasing exponentially. It means that every 8 years, we have twice as much, twice likely to die or get some very unlikely disease. And we see that all of them, Alzheimer, kidney disease, heart diseases, cancer, the probability of all these events grows with advancing age and grows rapidly. And even non-disease causes of dying, like accidents, are also, the chance of dying from an accident grows with advancing age. This is very sad, actually, yes. But this is like overall observation, like very broad picture. But what is aging on molecular or cellular level? Actually, there is very interesting paper which is called Hallmarks of Aging, and these things are happening with our body with advancing age. So our stem cells, which renew the population of different cells in our organism, the pools of these stem cells are exhausting with advanced age. We have cellular senescence. It means that some cells of our body become like senescent. They produce different chemical substances that harm our body. Our mitochondria, which are the power plants of our cells, their function now is improper. They produce less energy, and there are a lot of other consequences of that. So our proteins start to aggregate, and different epigenetic alterations, and so on. Genomic instability is increasing with advancing age, and it means that the probability of cancer also increases, because cancer is about mutations in our DNA, but not only, but this is one of the primary cases. And does it mean these scary things? Do they really mean that aging is inevitable? And to answer this question, let's face two facts. Maybe some of you heard about naked mole rat. Please, could someone raise hands if you heard about this tiny animal? Oh, I see. Some of you heard about it. Okay, here is this wonderful mammal. So why it is wonderful? Because some time ago, researchers thought that this animal doesn't age, and the lifespan of this animal is like about 30 years, which is quite high lifespan, because typically the body size correlates with longevity, and mice who are approximately that body size live about 4 years, and this naked mole rat lives about 30 years, and this is really nice. And but the more interesting thing that it is very, very cancer-resistant animal. So the prevalence of cancer is very, very low. Previously, it was stated that naked mole rat doesn't suffer from cancer. But now it is, there was some sad news about that scientists actually found some cancer incidents in naked mole rat. But nevertheless, the prevalence of cancer is extremely low, and this is really cool. But the more interesting is even more years of lifespan has another mammal, and it is Brand's bat, which is very, very tiny. And you see the correlation between body mass and lifespan, and there are two obvious outliers. We, human beings, and Brand's bat. And it lives about 42 years. But 42 years is very cool result in comparison with other species of that body mass, but we live much more than 42 years, so for us it's not like an outstanding result. But what about Greenland's shark? And it lives about 400 years. The estimate of this lifespan is rather inaccurate, but on average, it was estimated that lifespan is about 400 years. But nevertheless, this Greenland shark is experiencing signs of aging. So it ages, though slowly, but it ages nevertheless. But do we have any examples of non-senescent species? And the answer is yes. There is several, not only Hydra, but there are several species that do not age. Their mortality rates are constant at any age. So they are like biologically immortal. It means that if you will have in a cage or in a pool like millions of these hydras, they will live, at least some of them will live for any age that you want. Okay, but yes, so there is no law that says that we cannot, that biological species could not achieve like non-senescence. But could we, humans, influence this process? And the answer is yes and no, simultaneously. So this is typically how the scientists measure aging. They put some animals into the cage or like in the Petri plate, if we are talking about nematodes, and see. So at the start of the experiment, there are 100% of nematodes alive, and then with time, some of them start to die. And here, these curves represent the wild type nematodes. It is like small worms. And one mutation in only single gene made some of them very like centenarian make them live longer and become as centenarians in the world of worms. They lived, these DAF2 mutants lived 3 times longer than the wild type counterparts. And then, this is a remarkable result, but not the best. Actually, as you can see, we can extend the lifespan of worms 10 times. 10 times. It means that human could possibly live for 1,000 years if we can translate this result to human beings. And notice one important thing. This curve, it is called survival curve or Kaplan-Meier's curve, is the wild type curve, is much, much steeper. And this is a very important remark. So please remember that. But that was about genetic interventions. So we somehow introduced some mutations into the target genes, but the genetic therapy is not really available for each of us. So what about drugs? And the results are not so good, but nevertheless, the combination of some drugs extended lifespan of these worms by 100%. So they have twice longer lifespan. This is good, but what about humans? What about humans? Is it translatable for us? But the answer is, again, yes and no. So there is medication called statins. They are inhibitors of one enzyme which is involved in metabolism of lipids. And the combination of these statins and widely prescribed aspirin reduces our mortality at any point of life twice, reduces by 100%. So twice lower mortality. But what does it mean in terms of additional years of lifespan? I've made simple calculations, and it turns out that it gives us about 10 years of additional healthy life. So, do we really influence aging? And the answer is, again, yes and no. I've asked you to remember this difference in the slopes, and that's why. Here is the picture from the paper of Vladimir Anisimov, who is a pioneer of anti-aging research in Soviet Union and then in Russia. And here he describes 3 types of aging, of anti-aging drugs, substances. The first type is when we shift the survival curve to the right. And that means, actually, and here in the right corner we can see the incidence of cancer in mice. And here we see that cancer started, the probability of cancer started to grow after a while. So it somehow freezes, like aging process for a while, and then the aging process continues with the same rate. So we somehow stop aging for a while. Say, oh, hold on, aging, not yet. But then the aging continues to go with the same rate. The second type of these mortality curves is actually slow down aging, because we can see that the incidence of cancer is not only shifted on the time scale, but also it grows slowly. And the third type is when the incidence of age-related diseases, the point at the time when they start to occur, is shifted to the right, but the rate of increasing the probability of these diseases is higher. So it actually shifts a little bit, but then increases speed of developing these diseases. And this is third type of drug or intervention which extends life. And it turns out to be that we have written a book with Professor Moskalov. It is called Potential Geroprotectors. Geroprotectors is a drug which extends life. And it turns out to be that all of these drugs tested on mammals are these types. So they doesn't really slow down aging. They only could postpone it or postpone and then increase the rate of aging. And this is a big problem now. So we need, our best result is to stop aging, so to make the rate of aging to zero, to set it to zero. And for that we need these kind of interventions, but now we haven't that. Nevertheless, we have these types of interventions. And it's also good because it allows us to live 10 or even more years, because I know a couple of combinations that are more promising than a statin plus aspirin. And they can, in combination with healthy lifestyle, diets, physical activity, proper sleep, and many others, they can give like 10, 15, 20 years of additional healthy life until researchers will invent something. But how do we apply these interventions? How we see that this combination or this geroprotector or this kind of diet actually helps us? Of course, we will need to control our biomarkers. And my friend Stanislav will tell us a little bit more about these biomarkers, but I will shortly introduce some of concepts that I have. First of all, properties of ideal biomarker. It correlates with age. It predicts all-cause mortality, and it is robust or easy to measure. If the biomarker is robust, for example, we have very cool markers of inflammation. Inflammation is one hallmark of aging. But these markers like C-reactive protein or interleukin-6, they have high variability on the time scale. You can see that they varies greatly, the values of this. And so we need to measure them very frequently to see the picture, the trend. And that's why we need either robust markers that did not change so quickly, that don't change so quickly, or like non-invasive markers. And here are some examples. One of the markers that we, in our study, showed the best correlation and robustness is carotid intima media thickness. Carotid is an artery which feeds our brain. And that's why the carotid intima media thickness predicts incidence of dementia and cognitive impairment and many other things. Also, one of the robust markers is glycated hemoglobin. It is, we think that it correlates with blood glucose, but it reflects your average blood glucose level like for 1 to 3 months, so for like very large time period. But actually, it is a little bit different from blood glucose because it reflects the overall glycation process, which is one of the hallmarks of aging too. And it is a little bit separate from blood glucose, and it's better. Also, we have some biomarkers that you can measure every day and without any cost and without any invasive procedures. This is about your blood pressure and pulse. So your blood pressure reflects your arterial stiffness, your pulse pressure difference between systolic and diastolic blood pressure, and it's very informative and it's very predictive because it reflects the stiffness of arteries. With advancing age, our arteries become less elastic, and it is the cause of many diseases and cause of mortality. And also the very important and very easy to measure thing is resting pulse rate. Ideal value for resting pulse rate is 50 or less. And people with 60 have slightly increased mortality, but people with 90 plus pulse rate, resting pulse rate, have 3 times increased mortality from any cause. So this is very simple biomarkers that every of you can apply immediately and do it without any cost. So I think it was helpful for you, and thank you very much for your attention. That's all.